A warm welcome to everybody to this ISASO learning platform.
Hi, this is Giuseppe Querques. I wish to congratulate our friend Pippo Simona because of this little but at the same time huge work of interpretation of structural OCT. In these past years, we have seen a dramatic improvement in OCT imaging, and nowadays we face with a strict relationship between in vivo imaging with structural OCT and histopathology. Because of this, it is extremely important to understand this close relationship between in vivo OCT and histopathology. And this because may allow us to better understand diseases and better treat. This work by our friend Pippo Simona is not meant to make a new research, but it's extremely important because it is a strong recap on all the features that we need to take in consideration every time we are interpreting an OCT. And it's probably extremely useful not only for young ophthalmologists, for young retinal specialists, for residents, but also for skilled retinal specialists because of its important aim to make the point and to recap all the features. Congratulations again, people.
Those of you who have never had a small doubt about which retinal layer the deep vascular plexus runs into, cast the first stone. This brief compendium serves precisely as a grip to always have at hand to dispel any doubt. First of all, it is necessary to learn the famous 10 retinic layers by heart. Second step, it is very important to note that the 3 layers containing nerve cells appear gray. Step 3, the layers containing the nerve fibers are clear. Step 4, please note that the ellipsoidal line is bright white due to the high reflectivity of the mitochondria. Bruch's membrane is only 3 microns, but in the OCT it's like 15 microns. Here you see what the anatomy looks like, adapted to our OCT needs. Let's add now the histology. Take all the time you need to analyze the dense slide. In 2014, a large number of experts met and defined the exact nomenclature of each layer. This is a representation of the Heidelberg device. Here are the different layers of the Heidelberg manual. Here the different layers with the Zeiss device. We move now to the material that we have personally collected with our Cirrus device. This is the retina of our young optometrist. We now superimpose the macular anatomy of the fundus with the OCT image. First, we will have a representation of the ambo central of 150 microns. Then, the 350 microns fovea, then the fovea of 800 microns, then the parafovea with a diameter of 1800 microns, and finally the perifovea 5000 microns. And here the nomenclature and the OCT report. Let us now move on to the closer analysis of the individual layers. Here the retinal nerve fiber layer. In case of myelinated retinal nerve fibers, particularly well visible, like as in this patient from our consultation. Here, the appearance of the papilla in the Daytona picture. The thickness of the RNFL is measured automatically by the machine. The ganglion cell layer is immediately below. The quantification of the layer is automatic, and it is very effective for the diagnosis of multiple sclerosis. Here the quantification of the thickness of this layer in the macular zone, and here the thinning in a case of MS. The same thinning but in a case of glaucoma. The inner plexiform layer contains synapses between dendrites of ganglion cells, between bipolar and displeased amacrine cells, and between bipolar cells, ganglion cells, and some amacrine cells. Inner nuclear layer. And now the analysis of the outer retina. The outer plexiform layer, outer nuclear layer. Here the mirror cell is in yellow. The external limiting membrane, and a sketch of mine of a rabbit mirror cell. Here the myoid zone with the endoplasmatic reticulum and the ellipsoid zone with the mitochondria. Here the interdigitation zone where the sensory retina meets the EP. This wonderful electron microscope photo is essential. The EP microvilli wrap around the hand of the photoreceptors, but there is no close connection. This is where the detachment of the retina takes place. This virtual space is where retinal detachment happens. Carl Ludwig Wilhelm Bruch, 1819 till 1884. This new OCT allows you to take a more than detailed look at the choroid's structures. This is model 3D. In the macular region, there are no rods, but only cones, with a density such that the outer nuclear layer reaches its maximum thickness. The outer limiting membrane, in the absence of the other layers in the central area, forms a sort of depression called fovea externa. The layers of the inner retina reappear more laterally, welcoming the nerve fibers of the cones. The layer of the fibers of Henle starts laterally, assuming an horizontal course. Here the arrows to the different layers. The microscopic image of the fovea of this macaque illustrates the total absence of internal retinal layers in the foveolar zone.
It's really interesting to focus on the morphological development of the human fovea, because understanding the structure of the primate fovea can help to understand the beautiful mechanism underlying the human vision. Here you can see a model of foveal development. At 24th fetal week there is the presence of the area centralis with a domed ganglion cell layer. At 28th fetal week there is the presence of the foveal pit. By centrifugal displacement of the inner retinal layers, which increases the transparency of the central foveal tissue, the primate fovea improves the quality of the image received by the central photoreceptors. The foveal pit is proposed to be formed by a vertical contraction of the central Muller cells. After widening of the foveal pit, likely mediated by retracting astrocytes, Henle fibers are formed by horizontal contraction of Muller cell processes in the outer plexiform layer. At birth, the foveal pit is a deep bowl, but still all retinal layers are present in the center. And the central cones form a monolayer of cell somata in the outer nuclear layer. The foveola is formed after birth and the fovea externa after the 8th postnatal week. The foveola is formed by cone and Muller cells and is composed of 4 layers which lie in front of the fovea externa: the inner layer, the Henle fiber layer, the outer nuclear layer, and the layer of the outer cone cell processes. In addition to improving the optical properties of the foveola, the Muller cells may provide the structural stability of the central fovea. More than 90% of the cones in the human retina are red and green. The others are blue. A better understanding of the molecular, cellular, and mechanical factors involved in the developmental morphogenesis and the structural stabilization of the fovea may help to explain the pathogenesis of foveolar hypoplasia and macular holes.
And to finish, the retinal vascular plexus. Here, some interesting readings. It is important precisely at this time to support the close link between communication and training. We hope that you enjoy this presentation and that it will be useful to you.